there are now pretty reliable methodologies in the laboratory for growing very specific types of cells. So dopamine neurons that would be useful for Parkinson's disease, motor neurons that might be useful for motor neuron disease, types of inhibitory neurons that could treat epilepsy. So we can do all that, but that's all in the dish. And the real challenge in this field is how to introduce them into the brain and have them survive, connect, function properly, all that sort of thing. And that's where we come in, and that's our area of interest. If you want to look at a regenerative therapy, you have to have a model of the disease so that you know that the therapy is having an impact. We use a number of rat and mouse models that model specific aspects of human brain disease. So we work with, for example, a model of Parkinson's disease. And in Parkinson's disease, it's one particular cell type that dies. It's the dopamine neurons. And what we can do is introduce a toxin into the mice or rats that selectively kills those cells. And then they'll have a Parkinsonian phenotype. So they have deficiencies in motor behaviour, for example. So then you have a basis for testing your therapy. So we work with two types of stem cells. They're both what's called pluripotent. So they can make any type of cell from the organism from which they're derived. And we work with both embryonic stem cells and induced pluripotent stem cells. I guess the current thinking is that induced pluripotent stem cells will be, kind of the future of stem cell therapy, because you can establish them from the patient that you want to treat, right? So there'll be less complication with immune rejection and things like that. So the idea is that if you came along with a neurological condition that was something we could think about treating with cell therapy, then we would take a skin cell, for example. We would talk that skin cell into becoming pluripotent. And there's ways to do that in the laboratory. Normally you introduce genes, for example, these pluripotency genes. Then you would convince those pluripotent cells to become the therapeutic cell that you're interested in. And they're your cells, right? So you put them back in and you recognise them as your cells, you don't reject them. So that's the sort of the power of induced pluripotency. And one of the things we found is if you put these human cells in a mouse or rat, their growth pattern doesn't then just magically match what a normal rat and mouse does. It's still a human cell, right? So we found to see any benefit, you have to test these animals over sort of 6-9 months, a year. So we established for each animal a baseline set of data that tells us that animal's capacity for motor function. It's pretty non-invasive, it sounds nasty, but it's pretty straightforward. You introduce a toxin into the animal and it's very selective, so it only kills those neurons that you want to kill, which are the dopamine neurons. So that's a starting point. Then the next part is to grow the cells in the laboratory, and that's challenging in its own right, and that's something that the field's finding at the moment is what you do on one day in the laboratory doesn't necessarily equate to what you do the next week. So there's a lot of checks and balances to say, right, are we growing the right cell type? Do they look like the right cells for transplantation? Are they going to survive? And then you come to transplantation day, we transplant all the animals. And then the last piece in the puzzle is really to look at the post-mortem data. So the animals will come to histology, and then we can start to correlate the histological findings with the functional impact in each animal. So is it because certain transplants failed to wire up properly, certain transplants didn't survive? Is there a threshold number of neurons we need per transplant, things like that? That's really important because when we come to clinical trials, we'll know that, well, you need to put the transplant here or you need this many cells and so forth. What we found is that, like embryonic stem cells, neurons grown from induced pluripotent stem cells have a remarkable capacity for surviving after you transplant them in the brain and forming connections that are appropriate for the cell type that you sort of convince them to become before you put them in the brain. So that's, I guess, one of the main findings from our laboratory over the past 5 years or so is on the neuroanatomical side to show that these cells really can grow a very long distance in the brain.